So I'm going to kick off today, and I was asked by Richard to present an overview of what we've been discussing the past week. So this does come with a health warning. This is my reflections, not necessarily everybody that participated. So I'm going to apologize if I've missed anything or overseen anything. But after 4 days, my attention span is probably not as good as it should have been. So apologies if I've missed anything off. So first and foremost, Young Emerge. So they held their colloquia at the beginning of the week, 2 days from 52. This is actually, okay, never mind. I think it was about just over 44 attendees from UK, Europe, Australia, so across the globe, which is fantastic. Industry involvement as well. And the attendees covered everything from undergraduates through to MSc, PhD students and postdocs. So a whole span of the young investigators. The things they did, there was networking obviously, career development, progression, discussions, funding opportunities, collaborations, presentations on how to design a human study in the clinical trials, and of course being young ones, lots of social activities. So I think the pub in the village is really going to miss them. Their takings are going to plummet, I think, after the meeting. But overall, a very successful one. There's some very high quality, that should be early career research, ECR, science talks and posters, and there were prizes as well. So congratulations to those that won the oral presentation and the poster prizes. And the next Sparks workshop will be in Valencia in October 26th. So there's a clear momentum here, and this is really encouraging to see, because it's these guys that are the future of ME research. So, some key observations, and again, these are my observations, not necessarily reflective of everybody's. So, we are learning increasingly more in detail the key cellular players that we can identify that distinguish ME/CFS pathology, okay? And these are becoming increasingly uncovered through the use of sophisticated cutting-edge technology. These are not cheap, they're expensive, which obviously limits their application, but increasingly they're starting to be employed in ME/CFS. And the example I've given here is single-cell genomics, which can be looking at the transcriptome, the genome of an individual cell. So that's very exciting technology. Complexity and heterogeneity of the disease is now becoming increasingly apparent, and unsurprisingly for a lot of us, the genetics analysis that's been done, the GWAS studies, are reflecting the polygenic nature of the disease and how heterogeneous it is. We're also beginning to appreciate the need and the value for incorporating systems and network biology, which integrates into it artificial intelligence, machine learning. We're generating massive data sets. Somebody's got to figure out how we bring all these together and make sense of them. And this is where systems biology and networks biology come into the fore. And we're very fortunate that we have got some very skilled individuals that are contributing to ME/CFS research that are able to do this. We're also increasingly gaining insights from different fields. So we had quite a few presentations over the last 2 days from people focusing on long COVID. And as a lot of you know, some people that develop long COVID will have symptoms that would meet the diagnostic criteria for ME/CFS. So it's always interesting to learn what's going on with long COVID. And one of the key things there was the clinical and liquid tissue biomarker-based subtyping. So the blood is not necessarily the best place to look for evidence of the virus. We need to be looking elsewhere in the body, tissues, for example. And then we've learned how some viruses can actually subvert and overcome the host immune response to virus infections, which can contribute to the chronicity of infection that characterizes ME/CFS. And then we had some interesting talks on mitochondrial physiology coming from people that do detailed analysis of mitochondria down to the level of looking at what transporters are responsible for bringing in ATP, delivering it and exporting it. Again, some insights there that we can benefit from in ME/CFS. However, key challenges still remain. So because of the technology and the approach a lot of people are taking, it is still predominantly reductionist science. So we're drilling down on individual cells, okay? And in doing that, we just always have to be aware of the danger of losing the big picture, the context in which all these detailed cellular, molecular, genetic changes are occurring in individual cells. So we are the danger of not seeing the wood in the forest for the trees, and are we in fact looking in the right wood and the right forest? That's a word of caution, I think. Complexity and disease heterogeneity. So how many diseases, how many phenotypes make up ME/CFS? And should we even care about how many there are? Well, we don't know how many, but yes, we should care, because if we can accurately identify and subtype patients, we've got a much better chance of targeting effective therapies to those individuals. And this comes back to, or comes down to, the personalized precision medicine. We had some really interesting talks about deploying patient-centered approaches with all this high-resolution, fancy science that we have to do. And what we need to do is identify the individual signature of a patient, and that can get lost in all these large population studies. Because at the end of the day, we are going to be treating individual patients. So to know which patient can respond to the best drug is absolutely key. And we had a nice talk yesterday about GLP-1 receptor agonists and how that is being potentially useful in some, but not all, ME/CFS patients, using the power of genetics to identify the patients that may be best suited for that. So this, I think, will become more apparent over the next couple of years when all of this technology allows us to accurately subtype patients. And then multi-systems interactions and complexity. This is where we need a lot of expertise from across the whole panoply of biology. We need computational biologists, we need systems and network biology, we need AI, we need machine learning. All this comes at a cost in terms of, well, what do we do with the data? Where do we store it? How can we store it? So this is massive computing. And I think this is underappreciated. There are people in the audience that do this as their day job, and they are aware of the massive computational resource that we need. And this is often underappreciated. And then we still lack accurate and relevant disease models. There is no animal model for ME/CFS that would be informative in terms of investigating mechanism and drug therapy. So we need to move away from looking at individual cells to tissues and organs that reflect the physiology of those organ systems in ME/CFS patients. MPS is microphysiological systems that can preserve cellular architecture. And we've learned from individuals that are mapping or developing an atlas of every cell in the human body that in doing that, they're uncovering novel, unpredictable interaction between different cell types. So if once we break a tissue apart, we lose all of that. So it's important that we try and look at organs intact. And there are systems that are becoming available to enable to do that. And one of those is what we call organ-on-a-chip systems, but there are others as well. An important part is temporal dynamics, so we can get a snapshot of what's happening in a patient by taking one sample at one time point, but that tells us nothing about how that may change over the course of symptoms development, symptom progression, so we need to be doing longitudinal sampling. And that is from the whole body all the way down to genetic analysis. And the big question is causation or correlation, okay? What are the triggers? What causes ME/CFS? And again, a lot of the research we heard was looking forwards, so looking down different cell types, different organ systems, but we need to go back to say, okay, what's causing this? What is the initiating event that leads to this cascade of change in events that we're getting very, very good at defining and resolving? And I've just put up this immune dysfunction as an example of this. This primarily because this is one of my areas of interest. And again, I think how we look at immune dysfunction is reflective of this looking forward and not necessarily backward. So reductive forward approach is predominant. We're getting very, very good at defining individual cells, how they work or how they don't work, their phenotypes. So for the adaptive immune system, we're getting very good at describing changes in immune cell populations. But all this is occurring downstream of some trigger. So what's causing these changes? Reduced diversity of T-cell repertoires, B-cell repertoires, narrowing of antibody diversity. Is this driven by a specific antigen or antigens, or is it products of pathogens that are just left in the body that trigger these responses? We need to know more about the antigens, where they come from and what is their nature. And then in the innate immune system, there's been a lot of interest in this. So now we talk about NK cells, which have a long history of association with ME. Neutrophils, we had an interesting talk yesterday about neutrophils. And monocytes, and we've identified change in their epigenome, their metabolism, their survival program. And this may go all the way back to the bone marrow, which is the source of a lot of the progenitors of these cells. So these events may be happening very early in the immune system development. Triggers, well, there are obvious sources. There are things within the body and things outside the body, exogenous, endogenous. We need to know the relationship of these, because some of the viruses we get infected with can impact on the latent viruses that we harbor in our genomes and in our cells. So which pathogens could trigger reactivation? Some of the viruses, EBV, for example, that can lead to ME/CFS. And again, we've learned from other systems that things like influenza virus, SARS-CoV-2 viruses can reactivate the latent pathogens in the body. And these include enteroviruses. We had an interesting talk about enteroviruses may be underappreciated in terms of causing ME/CFS, maybe even pandemics. Herpes viruses, of course, Epstein-Barr virus, and endogenous retroviruses as well, which are part of our genome and have been with us since we've evolved. These can get reactivated in response to influenza infection, SARS-CoV-2, and these have been related to diseases like multiple sclerosis and cancer when they get activated. So we need to understand more about the relationship of exogenous and endogenous pathogens. And we now know they're hidden reservoirs of particularly viruses. They don't all live in the blood, they hide in tissues. And last year we published this review where we were proposing that these mucosal tissues or reservoirs of virus are underappreciated. We need to be able to start sampling these to identify where these viruses may actually hide and become active. And so what this cartoon shows is that in different mucosal tissues, they can harbor different viruses. So for example, the oral mucosa of Epstein-Barr virus. And these viruses can get activated by stress, another infection. They can replicate in these tissues. They can then escape from these tissues, get into the bloodstream, cause viremia, and all the inflammatory cascade that we associate with ME/CFS. And of course, this then leads to a lot of the symptoms. So, we need to pay more attention, I think, to where these reservoirs of viruses may hide. And then there are neglected, what I call pathobionts, but these could be pathogens, and fungi is one of these. Lots of patients we know report incidences of symptomology related to a fungal infection or a fungal infection occurring as a consequence of this. There are other pathogens as well, the archaea, these ancient bacteria, and protozoa as well, which could also be related to this. And then what about non-infectious triggers? We didn't really hear anything at the colloquium about triggers that are unrelated to pathogens, so stress, for example, environmental factors, toxins, these things. So that's another area of consideration. So I think that's all I want to say.